genes, and genetic diseases. DNA is made up of building blocks called nucleotides. Each nucleotide consists of three parts: a sugar called deoxyribose, a phosphate group, and a nitrogenous base. The bases can either be pyrimidines, like cytosine and thymine, or purines, like adenine and guanine. Together, these nucleotides arrange themselves into the iconic double helix structure, forming the foundation of all genetic material. This illustration highlights the structure of the DNA double helix. The two strands are twisted together like a spiral staircase, with base pairs connecting them in the center. Adenine always pairs with thymine, and cytosine always pairs with guanine. These base pairs are held together by hydrogen bonds, forming the rungs of the helix. This precise pairing ensures the accurate storage and transfer of genetic information. On the top left, we also see Watson and Crick, the scientists credited with describing the double helix model of DNA. DNA serves as the blueprint for protein synthesis. It directs the assembly of proteins, which are made up of one or more long chains called polypeptides. These polypeptides are built from smaller units called amino acids. There are 20 different amino acids used by the body, and the specific sequence of bases in the DNA, called codons, determines the order in which these amino acids are put together to form a protein. DNA replication begins with the untwisting and unzipping of the double helix. Each separated strand then serves as a template for creating a new complementary strand. DNA polymerase, an important enzyme, matches each base with its partner. Adenine pairs with thymine, and cytosine pairs with guanine. This precise base pairing ensures that the new DNA molecules are exact copies of the original. This illustration shows the process of DNA replication in action. The double helix unwinds, and each original strand serves as a template for building a new complementary strand. DNA polymerase matches each base precisely, pairing adenine with thymine and cytosine with guanine. This careful matching ensures the genetic code is accurately copied before the cell divides. The nucleus, where replication takes place, is shown at the top, emphasizing that this essential process occurs inside the cell's control center. A mutation is any inherited alteration of genetic material. It can involve large changes in the structure of chromosomes, called chromosome aberrations. One common type of mutation is a base pair substitution, where one base is swapped out for another. Another serious type is a frameshift mutation, which happens when a base is inserted or deleted. This shifts the entire reading frame of the genetic code, often resulting in major changes to the resulting protein. Sometimes mutations happen naturally without any outside cause. These are called spontaneous mutations. They occur in the absence of exposure to known mutagens like chemicals or radiation. Certain regions of chromosomes, called mutational hotspots, are more prone to changes. These hotspots have much higher mutation rates compared to other parts of the genetic material. Mutagens are agents that are known to increase the frequency of mutations. Some common mutagens include radiation, which can damage DNA directly, and chemicals, such as nitrogen mustard, vinyl chloride, alkylating agents, formaldehyde, and sodium nitrite. These agents can alter the structure of DNA, leading to changes that may be passed down through generations. Transcription is the process where RNA is made using DNA as a template. It begins when the enzyme RNA polymerase binds to a specific region of the DNA called the promoter site. This action starts the creation of messenger RNA, also known as mRNA. Once the mRNA strand is formed, RNA polymerase detaches from the DNA. The newly created mRNA then moves out of the nucleus and into the cytoplasm, where it will be used to build proteins. Transcription continues along the DNA until a special termination sequence signals it to stop. After transcription, the RNA molecule is not quite ready to be used. Many sections of the RNA, called introns, are removed. The remaining pieces, called exons, are then spliced together to create a final messenger RNA strand. Once splicing is complete, the mature mRNA leaves the nucleus and travels into the cytoplasm to guide protein synthesis. Translation is the process where RNA directs the synthesis of a polypeptide, which will become a protein. This happens through the interaction of messenger RNA with transfer RNA, also known as tRNA. The site where translation takes place is the ribosome. Each tRNA molecule carries a specific sequence of 3 nucleotides, called an anticodon, which pairs up perfectly with a matching 3 nucleotide codon on the messenger RNA strand. This precise pairing ensures that the correct amino acids are added to build the protein. During translation, the ribosome travels along the messenger RNA sequence, reading it 3 bases at a time. Each set of 3 bases corresponds to a specific amino acid, and the ribosome links these amino acids together in the correct order to build a polypeptide. This process continues until the ribosome reaches a special termination signal. Once the signal is reached, the completed polypeptide chain is released into the cytoplasm, ready to be folded into a functioning protein. This illustration shows the full journey from DNA to protein. It begins in the nucleus, where RNA polymerase reads the DNA and creates a strand of messenger RNA. After transcription, the RNA is processed and modified before leaving the nucleus. Once in the cytoplasm, the messenger RNA binds to a ribosome. Here, translation begins. Transfer RNA molecules bring amino acids to the ribosome, matching their anticodons to the codons on the messenger RNA. The ribosome links these amino acids together, forming a growing polypeptide chain, which will eventually fold into a functional protein. In the human body, there are 2 main types of cells when it comes to chromosomes. Somatic cells are the cells that make up most of our body tissues and organs. They contain 46 chromosomes, arranged in 23 pairs, and are called diploid cells. Somatic cells are formed through the process of mitosis. On the other hand, gametes are reproductive cells like sperm and eggs. They contain only 23 chromosomes, meaning they are haploid cells, with just 1 member from each chromosome pair. Gametes are created through a different process called meiosis. Chromosomes are divided into 2 main groups, autosomes and sex chromosomes. Autosomes are the first 22 pairs of chromosomes found in both males and females. Each pair is virtually identical and is described as homologous. The final pair are the sex chromosomes, which determine biological sex. In females, the sex chromosomes form a homologous pair, labeled XX. In males, they form a non-homologous pair, labeled XY. To visualize chromosomes, scientists create a karyotype, also known as a karyogram, which is an ordered display of all the chromosomes arranged by size and shape. A karyotype is a visual display of all the chromosomes in a cell, arranged in their matched pairs. On the left side of the image, you can see the 22 pairs of autosomes and 1 pair of sex chromosomes, organized by size and structure. On the right side, the diagram shows how homologous chromosomes replicate to form sister chromatids. Structures like kinetochores and cohesin proteins help hold the chromatids together and ensure proper separation during cell division. Karyotypes are an important tool for identifying genetic abnormalities. Euploid cells are cells that have a multiple of the normal number of chromosomes. Both haploid cells, which have one set of chromosomes, and diploid cells, which have two sets, are examples of euploid forms. However, when a euploid cell has more than the normal diploid number, it is called a polyploid cell. In triploidy, a zygote has 3 copies of each chromosome, for a total of 69 chromosomes. In tetraploidy, there are 4 copies of each chromosome, totaling 92. Sadly, neither triploid nor tetraploid fetuses are capable of surviving. Aneuploidy occurs when a somatic cell does not contain a multiple of 23 chromosomes. In this condition, a cell might have 3 copies of a single chromosome, a situation called trisomy. On the other hand, monosomy happens when there is only 1 copy of a chromosome instead of 2. Monosomy is usually lethal, but in some cases, infants can survive if they have trisomy involving certain chromosomes. Overall, when it comes to chromosome abnormalities, it is generally better to have extra genetic material than to have less. Non-disjunction is the most common cause of aneuploidy. It happens when homologous chromosomes or sister chromatids fail to separate properly during meiosis or mitosis. This mistake leads to cells with an abnormal number of chromosomes, which can result in genetic disorders like Down syndrome. This diagram shows how non-disjunction leads to abnormal chromosome numbers. Normally, during meiosis, chromosomes are supposed to separate evenly into new cells. But when non-disjunction occurs, chromosomes fail to separate properly during either the first or second division. As a result, some gametes end up with an extra chromosome, leading to a condition called trisomy, while others are missing a chromosome, resulting in monosomy. When these abnormal gametes are fertilized by a normal one, the resulting offspring will either have too many or too few chromosomes. Partial trisomy happens when only an extra portion of a chromosome, rather than a whole chromosome, is present in each cell. Another related condition is called chromosomal mosaicism. In this case, trisomy occurs, but only in some of the cells in the body, not all of them. Mosaic patterns can lead to milder or more varied symptoms, depending on how many cells are affected. Down syndrome is the best known example of aneuploidy and is caused by an extra copy of chromosome 21, a condition called trisomy 21. It occurs in about 1 out of every 800 live births. Individuals with Down syndrome often experience intellectual disability, along with physical features such as a low nasal bridge, epigenethal folds, a protruding tongue, low set ears, and poor muscle tone. The risk of having a child with Down syndrome increases significantly when the mother is older than 35 years. This image shows a child with Down syndrome, a genetic condition caused by trisomy 21. Many individuals with Down syndrome share common facial features such as a flat nasal bridge, upward slanting eyes with epigenethal folds, a protruding tongue, and low-set ears. Along with these physical characteristics, individuals may experience developmental delays and varying degrees of intellectual disability. Early intervention and supportive therapies can greatly enhance quality of life. One of the most common types of sex chromosome aneuploidy is trisomy X, where a female has 3 X chromosomes instead of 2. Symptoms can vary widely. Some individuals may experience sterility, menstrual irregularities, or intellectual disabilities. The effects tend to become more severe as the number of extra X chromosomes increases. Turner syndrome occurs in females who have only 1 X chromosome instead of 2. These individuals often have underdeveloped ovaries, leading to infertility. They typically have a short stature, around 4 feet, 7 inches tall. Other features include webbing of the neck, swelling known as edema, underdeveloped breasts with widely spaced nipples, and a high rate of fetal loss during pregnancy. In most cases, the single X chromosome is inherited from the mother. This slide shows two important aspects of Turner syndrome. On the left, we see a clinical image of a young girl with typical features of the condition, including short stature, broad chest, and swelling of the hands and feet. On the right, a karyotype confirms the diagnosis, showing the presence of only one X chromosome and the absence of a second sex chromosome. This genetic pattern is what leads to the distinctive physical characteristics and reproductive challenges seen in Turner syndrome. Klinefelter syndrome occurs in individuals who have at least two X chromosomes and one Y chromosome. Even though they have a male appearance, they may develop female-like breasts and have small testes, often resulting in infertility. Other features include sparse body hair and unusually long limbs. Some individuals have even more X chromosomes, such as XXY or XXXY patterns. The more X chromosomes present, the more severe the physical and developmental abnormalities tend to be. This image shows an individual with Klinefelter syndrome. Although the person has a male appearance, you can observe the development of female-like physical traits such as breast tissue and a rounded body shape. People with Klinefelter syndrome often have small testes, reduced body hair, and longer limbs compared to average males. These features become more noticeable as the number of extra X chromosomes increases. Sometimes chromosomes can break, and although the body has natural repair mechanisms, the healing process can sometimes alter the chromosome structure. These changes can lead to genetic abnormalities. Certain agents, called clastogens, increase the risk of chromosome breakage. Examples of clastogens include ionizing radiation, harmful chemicals, and some viruses. Deletion happens when there is a breakage or complete loss of a portion of DNA. One well-known example is cry du chat syndrome, which means cry of the cat in French. It is caused by the deletion of the short arm of chromosome number 5. Babies with this condition often have a distinct, high-pitched cry, along with low birth weight, severe intellectual disability, and a smaller than normal head size, known as microcephaly. This diagram shows how a deletion happens within a chromosome. A break occurs at 2 points along the chromosome, causing a segment, labeled here as N and O, to be lost. As a result, the chromosome becomes shorter, missing an important section of genetic material. This loss can disrupt the normal function of the genes and lead to various genetic disorders depending on what part of the chromosome is deleted. Duplication happens when a gene or a section of DNA is repeated within the chromosome. This type of abnormality usually causes less serious consequences than deletions, because it is generally better to have extra genetic material than to have missing pieces. For example, duplication in the same region affected in cry du chat syndrome can cause intellectual disability, but with fewer and less severe physical abnormalities compared to a deletion in that area. Inversions happen when there are 2 breaks in a chromosome and the piece between them flips around before reattaching. This results in a reversal of the gene order. For example, a sequence that originally reads ABCDEF might be rearranged to ABGFEJ. Inversions often occur during the natural repair process after a chromosome break. They can sometimes lead to a position effect, where the change in gene order alters how certain genes are expressed. Translocations happen when pieces of genetic material are exchanged between non-homologous chromosomes. In a reciprocal translocation, 2 different chromosomes break and swap segments, leading to an abnormal arrangement. In a Robertsonian translocation, the fusion occurs at the centromere, combining 2 chromosomes into one. These changes can disrupt gene function and sometimes lead to genetic diseases or disorders. This image compares a normal chromosome arrangement to a case of reciprocal translocation. On the left, we see 2 sets of normal chromosomes. On the right, a reciprocal translocation has occurred, where segments of two different chromosomes have been swapped. This exchange creates chromosomes with mixed genetic material, which can disrupt normal gene function and lead to genetic disorders depending on where the breaks and reattachments occur. Fragile sites are specific areas on chromosomes that tend to form breaks or gaps, especially when cells are grown in culture. One of the most well-known fragile site disorders is Fragile X syndrome. This condition is caused by a fragile site on the long arm of the X chromosome. It is associated with intellectual disability and is the second most common genetic cause after Down syndrome. Fragile X syndrome is more common in males because they have only one X chromosome, so any damage to that single X has a greater impact. A locus is the specific location or position of a gene along a chromosome. Each gene can come in different forms, known as alleles. An allele is a variation of a gene at a particular locus. When a locus has two or more common alleles that occur frequently in a population, it is called a polymorphism. These genetic variations contribute to the diversity we see in inherited traits. When both copies of a gene at the same location on a pair of chromosomes are identical, the individual is said to be homozygous for that gene. On the other hand, if the two gene copies are different at that same location, the person is heterozygous. These differences in gene pairing affect how traits are inherited and expressed. Genotype refers to what an individual has, the actual composition of their genes at a specific location on the chromosome. Phenotype, on the other hand, is what they show, the physical expression or characteristics that result from their genetic makeup. For example, an infant born with the genotype for PKU or phenylketonuria may develop intellectual disability if left untreated. However, if the condition is properly managed through diet, the child will appear outwardly normal, even though the genetic condition is still present. When two alleles are present at the same gene location, the dominant allele is the one whose effect is visible or observable. The recessive allele, by contrast, remains hidden unless both alleles are recessive. In genetics, we use capital letters to represent dominant alleles and lowercase letters to represent recessive ones. Some traits may also be codominant, where both alleles are expressed equally, rather than one being hidden. Single gene diseases can be passed down in several ways, depending on the mode of inheritance. These include autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive patterns. To better understand how these traits are passed through families, we use something called a pedigree. A pedigree is a chart that maps out inherited conditions across generations, usually starting with the proband, the first individual in the family diagnosed with the condition. In autosomal dominant inheritance, an affected child is typically the result of one parent who is affected and heterozygous for the trait, and another parent who is unaffected. The dominant allele may cause the disease, while the other allele may be normal. On average, half of the offspring will inherit the dominant allele and show symptoms of the disease, while the other half will be unaffected. This slide uses Punnett squares to illustrate how autosomal dominant traits are passed from parent to child. In the first chart, both parents are affected. If one parent carries two dominant alleles, labeled D, and the other is heterozygous, their child could inherit two dominant alleles and be severely affected, though this is rare. In the second chart, we see a more typical scenario where one parent is affected and the other is normal. Each child has a 50% chance of inheriting the dominant allele and expressing the disease, and a 50% chance of being completely unaffected. This pedigree chart shows how an autosomal dominant trait is passed down through generations. The shaded symbols represent individuals who are affected, and the unshaded ones are unaffected. Notice that the trait appears in every generation, and both males and females can inherit and transmit it. Because only one dominant allele is needed to express the trait, an affected parent has a 50% chance of passing it on to each child. Recurrence risk refers to the probability that a genetic disease will appear again in a family. For autosomal dominant traits, if one parent is affected and the other is unaffected, each child has a 50% chance of inheriting the disease-causing allele. It's important to remember that each birth is an independent event. That means the outcome of one child does not influence the chances for the next. Each has the same 1 in 2 risk of being affected. Penetrance refers to the percentage of individuals with a specific genetic makeup who actually show the expected physical traits or symptoms. Sometimes, a person may carry the gene for a disease but never develop the condition. This is called incomplete penetrance. A good example is retinoblastoma, an eye tumor seen in children. Although the gene is present, only about 90% of individuals with the mutation actually develop the disease. This pedigree chart illustrates incomplete penetrance. The red circles and squares represent individuals who have inherited the gene and show symptoms of the disease. However, notice the unshaded circle with an arrow pointing to it. This person carries the same gene but does not express the disease. This is a classic example of incomplete penetrance, where someone has the genotype but does not show the expected phenotype. Expressivity describes the range of signs and symptoms that can occur in different individuals who share the same genetic mutation. In other words, even when people have the same disease-causing gene, the way the condition appears can vary widely. A good example is neurofibromatosis type 1, where some individuals have just a few skin spots, while others may have tumors or serious complications. This variation can be influenced by modifier genes, environmental factors, or even different mutations at the same genetic location. Epigenetics refers to how the same DNA sequence can lead to different outcomes, depending on chemical modifications that affect how genes are turned on or off. These changes don't alter the genetic code itself, but can influence which genes are expressed. One example of this is genomic imprinting, where a gene from one parent is silenced or inactivated during transmission to the offspring. As a result, only the gene from the other parent is expressed, which can significantly impact the child's traits or risk for disease. With autosomal recessive inheritance, most affected individuals have parents who are both carriers, meaning they each carry one copy of the defective gene, but do not show symptoms themselves. For each pregnancy, there is a 25% chance that the child will inherit both copies of the recessive gene and be affected. This pattern affects males and females equally. A well-known example of an autosomal recessive disorder is cystic fibrosis. This slide shows both a pedigree chart and a Punnett square to illustrate autosomal recessive inheritance. In the pedigree, 2 affected children are shown in red, while their parents appear unaffected, indicating that they are likely carriers of the gene. The Punnett square below shows the genetic possibilities when both parents are carriers. There is a 25% chance the child will be affected, a 50% chance the child will be a carrier, and a 25% chance the child will inherit 2 normal genes and be unaffected. Consanguinity refers to the mating of 2 individuals who are closely related by blood. When this happens, the chance of both parents carrying the same recessive gene increases significantly. As a result, the recurrence risk for recessive disorders in their offspring becomes much higher. This is why genetic counseling is especially important in families where consanguineous relationships are present. Sex-linked conditions are genetic disorders caused by mutations on the sex chromosomes, most commonly the X chromosome. When a gene mutation is located on the X chromosome, it's referred to as X-linked. The majority of these disorders follow a recessive inheritance pattern. X-linked conditions are usually more commonly expressed in males because they have only one X chromosome. If that X carries the mutation, there is no second X to compensate for it, as there would be in females. In females, one of the two X chromosomes is permanently inactivated early in embryonic development. This process is called X inactivation, and it ensures that females, like males, have only one functional copy of the X chromosome in each body cell. Once a specific X chromosome is inactivated in a cell, all of that cell's descendants will inactivate the same X. This inactivation is random, fixed, and incomplete, meaning not all genes on the inactivated X are turned off completely. Biological sex is determined by the presence or absence of the SRY gene, which is located on the Y chromosome. The SRY gene acts as a master switch. When it is present, it triggers a series of genetic events that lead to male development. It influences the activity of many other genes located on different chromosomes, guiding the formation of male reproductive structures. Most X-linked disorders follow a recessive inheritance pattern. In these cases, affected males cannot pass the gene to their sons because sons inherit the Y chromosome from their fathers. However, all daughters of an affected male will inherit his X chromosome and become carriers. Female carriers, in turn, have a 50% chance of passing the gene to their sons who will be affected, and a 50% chance of passing it to their daughters who will become carriers. These Punnett squares show how X-linked traits are passed from parents to children. In the first example, an affected father and a carrier mother produce daughters who may be carriers and sons who may be affected. In the second example, when a carrier mother mates with a normal father, there is a 50% chance their sons will be affected and a 50% chance their daughters will become carriers. The third square shows that when both parents contribute an affected or carrier X chromosome, the chances for affected children increase. This helps explain why X-linked disorders are more common in males. Sex-limited traits occur only in one sex, usually because of anatomical or physiological differences between males and females. An example would be inherited ovarian or testicular defects, which naturally appear only in the sex that has those organs. Sex-influenced traits, on the other hand, can appear in both sexes, but are much more common in one than the other. Examples include male pattern baldness, which is far more common in men, and certain types of breast cancer that are more frequent in women, even though the genes exist in both sexes. During meiosis, a process called crossover occurs, where sections of DNA are exchanged between paired chromosomes. This can cause recombination of alleles that are located on the same chromosome. By studying how often recombination happens between two genes, scientists can estimate how far apart those genes are. Genes that are located close together are less likely to experience recombination and are said to be linked. This principle is the foundation for mapping the distance between genes on a chromosome. The Human Genome Project was a groundbreaking scientific effort that successfully mapped all the genes in human DNA. This massive achievement created a complete reference of where each gene is located and what it may control. As a result, researchers were able to identify genes linked to over 5,000 medical conditions, opening the door to better diagnosis, targeted treatments, and personalized medicine. A polygenic trait is a characteristic that is influenced by the combined effects of multiple genes, each contributing a small part to the overall outcome. A multifactorial trait, on the other hand, results from the combined influence of both genetics and environmental factors. These traits often have a threshold of liability, meaning that only when the combination of genetic and environmental influences crosses a certain point does the trait or condition actually appear. The recurrence risk for multifactorial diseases can vary significantly between different populations. In general, the more family members that are affected, the higher the recurrence risk becomes for others in the same family. Unlike single gene disorders, these risks are not calculated purely by genetics. Instead, they are determined empirically through direct observation and analysis of real-world data across generations.